The hidden failure inside neurons. Inside every neuron of the human brain runs a microscopic transport system that keeps the cell alive and connected to the rest of the nervous system. Long cylindrical structures known as microtubules extend through axons and dendrites, forming internal pathways that allow cellular cargo to travel across extraordinary distances. Motor proteins carry vesicles, signaling molecules, and mitochondria along these tracks, maintaining the flow of information that allows brain networks to coordinate thought, memory, and behavior. The stability of this transport system depends on a protein called tau. Tau attaches directly to microtubules and regulates their structure, keeping them stable enough to support long-distance transport while still allowing the dynamic changes required for neural plasticity. When this system functions normally, neurons can maintain complex connections across the brain for decades. But tau does not exist as a single uniform molecule. The human brain produces several different versions of tau through a process known as alternative gene splicing. These variations are called Tau isoforms. Each isoform interacts with microtubules in slightly different ways, altering how tightly the cytoskeleton of the neuron is stabilized. Under healthy conditions, neurons maintain a precise balance between these isoforms. This equilibrium allows microtubules to remain stable without becoming rigid, preserving the flexibility required for synaptic remodeling and neural adaptation. When this balance begins to shift, the internal architecture of the neuron changes. Certain isoforms become more abundant, altering how tau interacts with microtubules. At first, the neuron remains alive. Electrical signals can still travel across synapses. From the outside, brain function may appear largely intact. Yet inside the neuron, the transport system that supports those signals begins to slow. Cargo that once moved efficiently along axons now travels less reliably. Vesicles carrying neurotransmitters arrive at synaptic terminals with increasing delay. Mitochondria responsible for supplying energy to distant parts of the neuron accumulate unevenly along the cytoskeleton. The structural framework that once stabilized these transport routes gradually becomes less reliable. These changes do not immediately destroy neurons. Instead, they weaken the coordination between distant brain regions. Communication across neural networks begins slowing long before large-scale structural damage becomes visible on medical imaging. During this early stage, the first signs often appear as subtle changes in cognitive processing. Tasks that require planning or flexible decision-making begin taking longer to complete. Complex conversations demand greater concentration to follow. Shifting attention between multiple ideas becomes more difficult. Memory itself may remain largely intact. Familiar information can still be recalled, faces recognized, and routine activities performed without difficulty. What changes first is the speed at which different brain regions coordinate their activity. Because these early disruptions remain mild, they are often mistaken for fatigue, stress, or the normal effects of aging. Standard neurological examinations may detect little abnormality. Magnetic resonance imaging frequently reveals only minor variations in cortical structure that fall within the expected range for healthy individuals. Yet within the microscopic architecture of neurons, a molecular imbalance has already begun. The equilibrium between tau isoforms that once stabilized the microtubule network is shifting. This imbalance represents the earliest stage of a biological cascade that can gradually destabilize the structural framework of neurons throughout the brain. To understand how this process unfolds, it is necessary to examine the molecular architecture of tau itself and the mechanisms that regulate its different isoforms within the healthy nervous system. Biological architecture. Neurons maintain their structure and function through an internal framework known as the cytoskeleton. Unlike many other cell types, neurons must sustain extremely long cellular projections that extend far beyond the cell body. Axons can span distances of several centimeters within the brain and even longer within the spinal cord. These elongated structures require internal support systems capable of maintaining mechanical stability while simultaneously enabling continuous transport of cellular components along their length. The cytoskeleton consists of three major classes of protein filaments: microfilaments, intermediate filaments, and microtubules. Among these components, microtubules play the most critical role in maintaining axonal structure and supporting intracellular transport. Microtubules are hollow cylindrical polymers composed of repeating tubulin protein subunits. These polymers assemble into rigid tubular structures that extend throughout the axon and dendritic tree of the neuron. Microtubules function as both structural supports and transport pathways. Motor proteins such as kinesin and dynein attach to vesicles, mitochondria, and protein complexes and move along the microtubule surface. Kinesin typically carries cargo from the neuronal cell body toward the synaptic terminal, while dynein transports materials in the opposite direction. This bidirectional movement ensures that synaptic terminals receive the molecular components required for neurotransmission and that damaged cellular components can be returned to the cell body for recycling. However, microtubules are not permanently fixed structures. Tubulin subunits can assemble and disassemble dynamically, allowing the cytoskeleton to adapt to changing cellular conditions. This dynamic behavior allows neurons to adjust their internal architecture during processes such as synaptic plasticity, axonal growth, and structural remodeling. While this flexibility is necessary for normal neuronal function, excessive instability of microtubules would disrupt the structural integrity of the axon. To regulate this balance between flexibility and stability, neurons rely on a group of proteins known as microtubule-associated proteins. These proteins bind along the surface of microtubules and influence their assembly, spacing, and stability. Among these proteins, tau plays one of the most important roles in stabilizing the microtubule network within neurons. Tau proteins attach directly to the outer surface of microtubules through specialized binding domains located within their structure. When bound to microtubules, tau proteins promote the alignment of tubulin polymers and reduce the likelihood that tubulin subunits will detach. This stabilizing effect allows microtubules to maintain the long, continuous tracks required for axonal transport. In addition to stabilizing individual microtubules, tau helps organize microtubules into parallel bundles that strengthen the internal framework of the axon. The production of tau proteins is controlled by a gene known as MAPT, which stands for microtubule-associated protein tau. This gene contains multiple coding regions that can be combined in different ways during the processing of messenger RNA. Through a process known as alternative splicing, the MAPT gene generates several distinct versions of the tau protein. Each of these versions differs slightly in its molecular structure and its interaction with microtubules. In the adult human brain, 6 primary tau isoforms are produced through this splicing process. These isoforms vary in 2 major structural features. One feature involves the presence or absence of amino terminal inserts that influence how tau interacts with other cellular components. The second feature involves the number of microtubule binding repeat domains contained within the tau molecule. The repeat domains are particularly important because they determine how strongly tau binds to microtubules. Some tau isoforms contain 3 microtubule binding repeats, while others contain 4. These forms are commonly referred to as 3-repeat tau and 4-repeat tau, abbreviated as 3R-tau and 4R-tau. The additional repeat domain present in 4R-tau increases its binding affinity for microtubules, allowing it to stabilize the cytoskeleton more strongly than 3R-tau. In a healthy adult brain, neurons maintain a balanced ratio between 3R and 4R-tau isoforms. In the adult human brain, neurons typically maintain a near-balanced ratio between 3R and 4R-tau isoforms through regulated splicing of the MAPT gene. This equilibrium ensures that microtubules remain stable enough to support long-distance axonal transport. while still retaining the flexibility required for structural adjustments during neuronal activity. Different regions of the brain may express slightly different proportions of tau isoforms depending on the functional demands placed on their neurons. However, the overall balance between 3R and 4R tau remains tightly regulated across the nervous system. Maintaining this balance is essential for preserving the stability of the microtubule network. In addition to isoform variation, tau proteins undergo chemical modifications that regulate their interaction with microtubules. One of the most important of these modifications is phosphorylation. Phosphorylation occurs when phosphate groups are attached to specific amino acid residues within the tau molecule. This modification alters the electrical charge and three-dimensional structure of the protein. Under normal conditions, moderate phosphorylation allows tau to detach temporarily from microtubules when structural reorganization is required. Enzymes known as kinases add phosphate groups to tau, while phosphatases remove them to restore the original structure of the protein. The balance between these opposing enzymatic activities ensures that tau proteins bind and release microtubules in a controlled manner. Through the combined regulation of alternative splicing and phosphorylation, neurons maintain a highly dynamic yet stable cytoskeletal system. Microtubules remain strong enough to support long axonal projections while still adapting to changes in neuronal activity. This finely tuned system depends on the precise balance between tau isoforms generated through MAPT gene splicing. When this equilibrium becomes disrupted, the ability of tau to regulate microtubule stability begins to change. The earliest stage of tau-related neurodegeneration begins when alterations in this isoform balance destabilize the microtubule network that supports neuronal architecture. Early mechanism. The earliest biological trigger of tau-related neurodegeneration begins at the level of gene regulation within neurons. The MAPT gene produces tau proteins through a process known as alternative splicing, in which different segments of the gene are selectively included or excluded during the formation of messenger RNA. This process determines which tau isoforms will be synthesized by the cell. Under normal physiological conditions, the regulatory proteins controlling this splicing process maintain a balanced production of 3-repeat and 4-repeat tau isoforms. A critical element in this regulatory system involves the inclusion or exclusion of a genetic segment, known as Exon 10 within the MAPT transcript. When Exon 10 is excluded during RNA processing, the resulting tau molecule contains 3 microtubule binding repeat domains and forms the 3R tau isoform. When Exon 10 is included, the tau molecule contains 4 repeat domains and becomes the 4R isoform. The precise regulation of exon 10 splicing determines the ratio between these two forms. In healthy neurons, splicing factors interact with regulatory sequences surrounding exon 10 to ensure that inclusion and exclusion occur at roughly equal rates. This regulation maintains a near 1-to-1 ratio between 3R and 4R tau isoforms. The balanced presence of these proteins allows microtubules to remain stable while preserving the dynamic flexibility required for structural remodeling within the neuron. When this splicing regulation becomes disrupted, the equilibrium between tau isoforms begins shifting. Mutations within the MAPT gene, alterations in splicing regulatory proteins, or changes in intracellular signaling pathways can influence the probability that exon 10 will be included in the final transcript. Even subtle changes in this probability can produce measurable shifts in the relative abundance of 3R and 4R tau proteins within neurons. An excess of 4R tau increases the overall microtubule binding affinity of tau proteins. While this might initially appear to enhance structural stability, excessive microtubule binding reduces the flexibility required for normal cytoskeletal dynamics. Microtubules must undergo continuous remodeling to accommodate axonal transport, synaptic plasticity, and metabolic changes. When tau binds too strongly, the cytoskeleton becomes excessively rigid, interfering with these dynamic processes. Conversely, an excess of 3R tau weakens the overall stability of microtubules. Because 3R tau binds less tightly to the tubulin lattice, microtubules become more susceptible to depolymerization. Under conditions of metabolic stress or mechanical strain, segments of the microtubule network may disassemble more easily. This instability compromises the structural support required to maintain axonal integrity. Both forms of imbalance disrupt the structural equilibrium of the neuronal cytoskeleton. The axon relies on stable microtubule bundles to maintain its elongated shape and to support the transport systems that deliver cellular materials to distant synaptic terminals. When microtubule stability becomes compromised, the organization of these bundles begins changing. Microtubules within the axon may become irregularly spaced or partially fragmented. Motor proteins traveling along these tracks encounter interruptions where the structural continuity of the microtubule network has been disrupted. As a result, the efficiency of axonal transport begins declining. Vesicles carrying neurotransmitters, enzymes, and structural proteins move more slowly along the axon, and some cargo may accumulate behind damaged segments of the cytoskeleton. Mitochondrial distribution within the neuron also becomes affected. Mitochondria must travel along microtubule pathways to supply energy to regions of the neuron with high metabolic demand. Synaptic terminals, in particular, require constant ATP production to support neurotransmitter release and membrane potential maintenance. When microtubule instability interferes with mitochondrial transport, these terminals may experience localized energy shortages. At the same time, tau proteins themselves begin exhibiting altered intracellular distribution. Under normal conditions, tau is concentrated primarily within axons, where microtubule stabilization is most critical. However, when isoform imbalance weakens the interaction between tau and microtubules, more tau molecules detach from the cytoskeleton. These detached molecules accumulate within the cytoplasm and may begin appearing in cellular compartments where tau is normally scarce. The increasing concentration of free tau proteins inside the neuron creates conditions that favor abnormal protein interactions. Tau molecules possess regions capable of interacting with other tau proteins when exposed within the cytoplasm. Under normal conditions, these interactions remain limited because most tau molecules are bound to microtubules. When large numbers of tau proteins become detached, the likelihood of intermolecular interaction increases. Initially, these interactions produce small soluble assemblies composed of several tau molecules. These early aggregates remain relatively unstable and dispersed within the cytoplasm. However, their formation represents the first step toward the development of larger pathological tau structures. Meanwhile, the destabilization of microtubules continues affecting the internal organization of the neuron. Axonal transport becomes progressively less efficient. Synaptic terminals receive fewer molecular resources, and mitochondrial distribution becomes uneven. Although the neuron may remain alive and capable of transmitting electrical signals during this stage, its structural resilience has begun declining. This early mechanism establishes the molecular environment that allows tau pathology to escalate. As microtubule instability increases and free tau molecules accumulate within the cytoplasm, additional biochemical processes begin altering the structure of tau proteins themselves. These modifications further weaken the interaction between tau and microtubules and promote the formation of increasingly stable tau aggregates within the neuron. Escalation. As the imbalance between tau isoforms destabilizes the microtubule network, additional biochemical processes begin amplifying the disruption inside neurons. One of the most significant changes during this stage involves abnormal phosphorylation of tau proteins. Phosphorylation is a chemical modification in which enzymes attach phosphate groups, specific amino acids, within a protein. Under normal conditions, phosphorylation regulates how strongly tau binds to microtubules and allows neurons to adjust cytoskeletal stability in response to metabolic or synaptic activity. In healthy neurons, phosphorylation and dephosphorylation remain tightly balanced. Kinase enzymes add phosphate groups to tau at controlled rates, while phosphatase enzymes remove these modifications to restore the original structure of the protein. This cycle ensures that tau can detach briefly from microtubules when structural reorganization is required, and then return to maintain stability once the adjustment is complete. When cytoskeletal instability develops, several signaling pathways may alter the activity of these enzymes. Kinases responsible for phosphorylating tau become more active under conditions of neuronal stress. Among the most influential of these enzymes are glycogen synthase kinase 3-beta and cyclin-dependent kinase 5. Both enzymes target specific phosphorylation sites within the tau molecule that regulate its interaction with microtubules. As kinase activity increases, tau proteins accumulate phosphate groups at multiple locations along their structure. Each phosphate group adds a negative electrical charge to the protein, altering its three-dimensional conformation. These structural changes reduce the ability of tau to bind tightly to the surface of microtubules. The stabilizing effect that tau normally provides begins diminishing as increasing numbers of molecules detach from the cytoskeleton. The detachment of tau from microtubules produces two major consequences inside the neuron. The first involves further destabilization of the microtubule network. Without sufficient tau molecules reinforcing the tubulin polymers, microtubules become more vulnerable to depolymerization. Segments of the cytoskeleton begin disassembling under normal cellular stress, weakening the structural support that maintains the shape of axons and dendrites. The second consequence involves the rapid accumulation of soluble tau proteins within the neuronal cytoplasm. Hyperphosphorylated tau molecules remain detached from microtubules and begin circulating freely within the intracellular environment. As the concentration of free tau increases, the likelihood of abnormal interactions between tau molecules rises significantly. These interactions occur because hyperphosphorylated tau exposes regions of its structure that normally remain concealed when bound to microtubules. The exposure of these regions allows tau molecules to interact with one another through intermolecular binding. At first, these interactions produce small soluble oligomers consisting of several tau molecules clustered together. While these oligomers remain smaller than the large aggregates that appear later in disease progression, they already interfere with normal cellular processes. Soluble tau oligomers can disrupt the organization of microtubules by interacting with tubulin polymers in ways that prevent proper alignment. This interaction further destabilizes the cytoskeleton and accelerates microtubule disassembly. Meanwhile, the loss of stable microtubule tracks begins severely affecting axonal transport systems. Motor proteins traveling along the axon encounter increasing numbers of interruptions where microtubules have fragmented or become misaligned. Vesicles containing neurotransmitters accumulate behind these structural barriers because they cannot continue their journey toward synaptic terminals. Mitochondrial transport also becomes disrupted during this stage. Mitochondria must be distributed throughout the neuron to provide ATP for energy-demanding processes such as synaptic transmission and membrane ion regulation. When microtubule tracks deteriorate, mitochondria cluster near the cell body and fail to reach distal regions of the axon and dendrites. These distant regions begin experiencing localized energy deficits. Energy shortages within synaptic terminals have immediate functional consequences. The release of neurotransmitters requires continuous ATP supply to maintain vesicle recycling and membrane ion gradients. When mitochondria cannot reach these regions efficiently, synaptic signaling becomes less reliable. Communication between neurons begins weakening even before structural degeneration becomes extensive. Hyperphosphorylated tau molecules may also appear in parts of the neuron where they are normally absent. In healthy neurons, Tau remains concentrated primarily within axons. However, abnormal phosphorylation alters its intracellular distribution. Some tau molecules begin accumulating within dendrites and near synaptic membranes, interfering with receptor signaling and synaptic organization. These combined effects create a self-reinforcing cycle of cellular stress. Microtubule destabilization disrupts axonal transport, while impaired transport reduces the delivery of mitochondria and structural proteins needed to maintain cytoskeletal stability. The resulting metabolic stress increases kinase activity, leading to further phosphorylation of tau and additional detachment from microtubules. As hyperphosphorylated tau accumulates within the cytoplasm, the small oligomeric clusters formed during early stages begin undergoing structural rearrangements that promote stronger intermolecular binding. These rearrangements allow tau molecules to assemble into filamentous structures capable of growing progressively, larger inside the neuron. The escalation phase, therefore, marks the transition from subtle cytoskeletal instability to widespread disruption of intracellular transport and protein organization. The accumulation of hyperphosphorylated tau molecules creates the molecular environment necessary for the formation of large tau aggregates that will soon begin interfering with multiple cellular systems simultaneously. Cellular damage. As hyperphosphorylated tau accumulates within the neural cytoplasm, the small soluble oligomers formed during the escalation phase begin transforming into more organized filamentous structures. This transformation occurs when individual tau molecules align along shared structural regions that allow stronger intermolecular binding. The alignment of these molecules produces elongated filaments composed of stacked tau proteins arranged in repeating patterns. One of the most characteristic structures produced during this process is the paired helical filament. In this configuration, two strands of tau molecules twist around one another in a helical pattern. The repeating twists stabilize the filament and allow it to extend as additional tau molecules attach to its growing ends. These filaments represent structural components of the intracellular aggregates associated with advanced tau pathology. As paired helical filaments accumulate, they begin associating with one another to form dense bundles within the neuronal cytoplasm. These bundles gradually grow into structures known as neurofibrillary tangles. Neurofibrillary tangles consist of large networks of aggregated tau filaments that occupy significant portions of the neuron's internal space. Unlike soluble tau proteins that once stabilized microtubules, these aggregated forms no longer support the cytoskeleton. The formation of neurofibrillary tangles produces immediate mechanical and biochemical disruptions inside the neuron. The dense filament networks interfere with the normal organization of cellular organelles. Mitochondria, vesicles, and protein complexes that normally move freely within the cytoplasm encounter physical obstacles created by these aggregates. The crowded intracellular environment slows the movement of materials required for normal cellular function. Axonal transport becomes increasingly impaired as tau aggregates expand. Motor proteins responsible for transporting vesicles along microtubules encounter both the destabilized cytoskeleton and the physical obstruction created by tau filaments. Cargo transported along the axon begins accumulating behind these blockages, forming clusters of vesicles and organelles within swollen regions of the axonal shaft. These swollen regions, often described as axonal spheroids, indicate areas where the internal architecture of the axon has collapsed. Vesicles containing neurotransmitters accumulate within these regions because they cannot reach synaptic terminals efficiently. The swelling distorts the axonal membrane and increases mechanical stress on the surrounding cytoskeletal elements. Mitochondrial distribution also becomes severely disrupted. Neurons require mitochondria throughout their axons and dendrites to provide ATP for energy-demanding processes such as synaptic transmission and ion regulation. When tau aggregates obstruct intracellular transport, mitochondria accumulate near the cell body instead of reaching distant synaptic terminals. These terminals experience localized energy deficits as a result. Energy shortages affect several essential neuronal functions. Ion pumps that maintain membrane potential require continuous ATP to regulate sodium and potassium gradients across the cell membrane. When energy supply declines, these pumps operate less efficiently, allowing ions to accumulate inside the neuron. Increased intracellular calcium levels activate enzymes capable of degrading structural proteins within the cytoskeleton. Calcium-activated proteases begin breaking down proteins that maintain the integrity of microtubules and other cytoskeletal components. This degradation further weakens the structural stability of axons and dendrites. As the cytoskeleton deteriorates, the neuron becomes more vulnerable to mechanical strain and metabolic stress. Tau aggregates also interfere with the cellular systems responsible for removing damaged proteins. Under normal conditions, misfolded proteins are tagged with small molecules called ubiquitin and directed to the proteasome for degradation. However, the filamentous structure of tau aggregates makes them difficult for the proteasome to process. As these aggregates accumulate, they overwhelm the capacity of the protein degradation system. Autophagy pathways, which normally remove larger cellular structures through lysosomal digestion, also become impaired. The size and density of neurofibrillary tangles often exceed the ability of autophagic vesicles to engulf them efficiently. As a result, tau aggregates persist inside neurons and continue disrupting intracellular organization. The presence of tau aggregates also affects dendritic structure. Dendritic spines, which serve as the primary sites of synaptic communication between neurons, rely on a stable cytoskeleton to maintain their shape and receptor organization. When tau pathology spreads into dendritic compartments, the structural proteins supporting these spines begin deteriorating. Dendritic spines gradually shrink and disappear as cytoskeletal stability declines. Each lost spine represents the removal of a synaptic connection between neurons. The cumulative loss of thousands of such connections significantly reduces the ability of neurons to receive signals from surrounding neural circuits. As synaptic connections disappear, communication within the surrounding neural network weakens. Neurons that once participated in densely interconnected circuits become progressively isolated as their synaptic contacts decline. Meanwhile, the axon continues experiencing structural deterioration due to the combined effects of microtubule instability and tau aggregation. Transport systems remain obstructed, mitochondria remain unevenly distributed, and cytoskeletal proteins continue degrading under the influence of calcium-activated enzymes. Eventually, sections of the axon may fragment entirely. When cytoskeletal support collapses beyond repair, segments of the axon detach from the neuronal cell body and degenerate. Once these segments disappear, the connection between neurons is permanently lost. The formation of neurofibrillary tangles therefore represents more than the accumulation of abnormal proteins. These aggregates disrupt multiple cellular systems simultaneously. They destabilize the cytoskeleton, block axonal transport, impair mitochondrial distribution, and eliminate synaptic connections. At this stage, the damage extends beyond individual neurons. The cellular stress produced by tau aggregation begins activating immune responses within surrounding neural tissue, initiating processes that will further amplify the degenerative cascade. Immune/metabolic amplification. As tau aggregation expands within neurons, the surrounding neural environment begins responding to the presence of damaged cellular structures and abnormal protein fragments. The central nervous system contains specialized immune cells, known as microglia, that continuously monitor neural tissue for signs of injury or molecular disruption. When neurons experience cytoskeletal collapse, synaptic loss, or protein aggregation, they release molecular signals that activate this immune surveillance system. Fragments of misfolded tau, damaged membrane components, and molecules associated with cellular stress enter the extracellular environment as neurons attempt to manage intracellular damage. These molecules function as danger-associated signals that attract nearby microglial cells. Microglia detect these signals through pattern recognition receptors located on their cell surface. Activation of these receptors initiates a transformation in microglial behavior. In their resting state, microglia extend long processes that scan the surrounding neural tissue while maintaining minimal inflammatory activity. When activated by molecular signals from damaged neurons, these cells retract their processes and adopt a more compact morphology associated with immune activation. Activated microglia begin producing inflammatory cytokines intended to coordinate tissue repair and remove cellular debris. Cytokines such as interleukin-1 beta, tumor necrosis factor alpha, and interleukin-6 accumulate within the extracellular space surrounding affected neurons. These molecules influence multiple signaling pathways within neighboring cells. Although their initial role is to facilitate clearance of damaged cellular components, prolonged exposure to these inflammatory signals begins altering neuronal metabolism and cytoskeletal regulation. One of the key interactions during this stage involves the effect of inflammatory cytokines on tau phosphorylation. Cytokine signaling stimulates intracellular pathways that increase the activity of kinase enzymes responsible for modifying tau proteins. Enzymes such as glycogen synthase kinase, 3-beta, and cyclin-dependent kinase 5 become more active in response to inflammatory signaling. As these kinases phosphorylate tau at additional sites, the detachment of tau from microtubules accelerates. This biochemical interaction establishes a feedback loop between neuroinflammation and tau pathology. Increased phosphorylation produces more soluble tau molecules capable of forming aggregates. These aggregates cause additional neuronal stress, releasing further molecular signals that sustain microglial activation. The inflammatory response therefore becomes self-amplifying as tau pathology progresses. Astrocytes also contribute to this amplification process. Astrocytes normally regulate extracellular ion concentrations, recycle neurotransmitters, and provide metabolic support to neurons. When exposed to inflammatory cytokines released by activated microglia, astrocytes adopt a reactive phenotype characterized by increased production of inflammatory mediators. Reactive astrocytes release chemokines that attract additional microglial cells and enhance local immune activity. This inflammatory environment alters the metabolic conditions within neural tissue. Activated microglia generate reactive oxygen species as part of their immune response. These molecules contain unstable oxygen atoms capable of reacting with proteins, lipids, and DNA. While reactive oxygen species can destroy pathogens during acute immune reactions, their presence in neural tissue exposes neurons to oxidative stress. Oxidative stress affects several critical cellular systems. Lipid membranes within neurons are particularly vulnerable because they contain unsaturated fatty acids that readily undergo chemical modification when exposed to reactive oxygen molecules. Oxidative damage to these membranes disrupts ion channel function and alters the electrical properties of neuronal membranes. Mitochondria also experience damage during prolonged oxidative stress. These organelles generate ATP through oxidative phosphorylation within the electron transport chain. Reactive oxygen molecules can disrupt components of this chain, reducing the efficiency of energy production. As mitochondrial function declines, neurons experience increasing difficulty generating the ATP required to sustain normal cellular activity. Energy deficits have immediate consequences for neuronal survival. Maintaining membrane potential requires continuous operation of ion pumps that regulate sodium and potassium gradients across the cell membrane. When ATP supply declines, these pumps begin failing. Sodium ions accumulate inside the neuron, and calcium regulation becomes disrupted. Elevated intracellular calcium activates enzymes that degrade structural proteins within the cytoskeleton. Proteases begin breaking down microtubule-associated proteins and other components required to maintain cellular integrity. These enzymatic reactions accelerate the structural deterioration already caused by tau aggregation. At the same time, reduced energy availability impairs the cellular systems responsible for removing misfolded proteins. Proteasomal degradation and autophagic clearance both require significant ATP to function effectively. When mitochondrial output declines, these protein clearance mechanisms operate less efficiently. Tau aggregates that might otherwise be degraded remain inside neurons and continue expanding. The inflammatory environment also influences how tau pathology spreads between neurons. Small tau aggregates released from damaged neurons can enter the extracellular space and interact with neighboring cells. Under certain conditions, these aggregates are taken up by nearby neurons through endocytic pathways or synaptic transmission mechanisms. Once inside a new neuron, these aggregates can induce misfolding of normal tau proteins through templated interactions. The abnormal structure of the aggregate promotes the conversion of soluble tau into additional filamentous forms. This mechanism allows tau pathology to propagate along synaptically connected neural networks. Microglial activity may facilitate this propagation by transporting tau fragments while clearing extracellular debris. The immune system, therefore, becomes indirectly involved in distributing pathological tau species throughout affected brain regions. Through the combined effects of neuroinflammation, oxidative stress, mitochondrial dysfunction, and impaired protein clearance, tau pathology transitions into a self-sustaining degenerative process. The molecular imbalance that originally destabilized microtubules has now expanded into a widespread disturbance, affecting multiple cellular systems. As these processes continue, structural damage accumulates within neurons and their connections. The next phase of degeneration involves the physical breakdown of neuronal architecture as synaptic connections disappear. Axons fragment, and entire neural circuits begin losing their structural organization. Structural destruction. As tau aggregation, inflammatory signaling, and metabolic stress continue interacting inside neural tissue, the structural architecture of neurons begins undergoing progressive deterioration. The cytoskeletal framework that once maintained the shape of axons and dendrites gradually loses its integrity. Microtubules that previously formed stable transport pathways disassemble under the combined influence of tau detachment, enzymatic degradation, and oxidative damage. Without these structural supports, the long projections of neurons become increasingly unstable. One of the earliest visible consequences of this structural decline occurs within dendritic spines. Dendritic spines are small protrusions extending from dendrites that serve as the primary sites of excitatory synaptic communication. Each spine contains a specialized cytoskeletal network that supports clusters of neurotransmitter receptors and signaling proteins required for synaptic transmission. When tau pathology disrupts cytoskeletal stability, this delicate structural system begins deteriorating. The destabilization of cytoskeletal proteins causes dendritic spines to shrink and eventually disappear. As spine density declines, neurons lose the physical structures required to receive synaptic input from neighboring cells. Because a single neuron may contain thousands of dendritic spines, even moderate reductions in spine density significantly weaken the connectivity of neural circuits. The loss of dendritic spines also disrupts the distribution of neurotransmitter receptors across the dendritic membrane. Receptor proteins that normally concentrate within spine heads become dispersed or degraded as the supporting cytoskeleton collapses. Without these receptors properly positioned, incoming synaptic signals cannot be transmitted efficiently into the neuron. Dendritic branches themselves also become affected as cytoskeletal instability spreads. Microtubule bundles that run through dendrites provide mechanical support and serve as tracks for transporting proteins required for synaptic maintenance. When tau aggregation and proteolytic enzymes disrupt these microtubules, sections of the dendrite may thin or retract. This retraction reduces the number of synaptic contacts that the neuron can maintain. Axons experience similar structural deterioration. The axon depends heavily on microtubule bundles to maintain its elongated structure and to support long-distance transport of vesicles and organelles. As tau pathology destabilizes these microtubules, axonal transport systems become severely impaired. Vesicles and mitochondria accumulate behind damaged sections of the cytoskeleton, producing localized swellings along the axonal shaft. These swellings, known as axonal spheroids, represent areas where the internal architecture of the axon has collapsed. Accumulated vesicles, damaged mitochondria, and fragmented cytoskeletal proteins fill these regions because transport pathways are blocked. The swelling places additional mechanical stress on the surrounding axonal membrane, further weakening the structural stability of the fiber. As the cytoskeleton continues deteriorating, segments of the axon may undergo fragmentation. Proteolytic enzymes activated by elevated calcium levels degrade structural proteins that maintain the continuity of the axonal fiber. Once these proteins are destroyed, the axon separates into disconnected segments. This fragmentation interrupts the pathway required for electrical signals to travel between neurons. Microglial cells rapidly respond to these degenerating structures. They migrate toward damaged axons and dendrites and begin engulfing cellular debris through phagocytosis. Although this process removes damaged material that could otherwise interfere with surrounding tissue, it also eliminates the structural remnants required for reconnecting severed neural pathways. Within the neuronal cell body, the accumulation of neurofibrillary tangles begins interfering with the organization of internal organelles. Tau filaments occupy increasing portions of the cytoplasm, compressing mitochondria and disrupting the distribution of ribosomes responsible for protein synthesis. The crowded intracellular environment further reduces the neuron's ability to maintain normal metabolic activity. Mitochondrial dysfunction intensifies during this stage. Damaged mitochondria release additional reactive oxygen species into the cytoplasm, increasing oxidative stress within the neuron. The resulting chemical damage affects proteins involved in maintaining membrane stability and ion channel function. At the tissue level, the cumulative loss of dendrites, synapses, and axons begins producing visible structural changes in the brain. Cortical layers that once contained densely interconnected neuronal networks gradually thin as synaptic connections disappear. Although many neurons may still remain present during this stage, the complexity of their interconnections has been significantly reduced. The hippocampus is particularly vulnerable to this form of structural destruction. Hippocampal neurons rely heavily on stable microtubule networks to maintain long axonal projections and elaborate dendritic trees. As tau pathology spreads through hippocampal circuits, synaptic connections between neurons weaken, reducing the structural integrity of the network responsible for memory formation. Cortical regions involved in executive function and behavioral regulation also undergo similar structural changes. Neurons within the frontal cortex lose dendritic spines and axonal connections, reducing the complexity of circuits responsible for planning and decision-making. Although electrical signaling may still occur in surviving neurons, the structural network required for coordinated activity becomes progressively fragmented. White matter pathways connecting distant brain regions begin, showing signs of degeneration as axons fragment and disappear. These long-distance fibers once allowed rapid communication between cortical and subcortical structures. When these pathways deteriorate, signals traveling between regions encounter interruptions that slow or completely block communication. The progressive breakdown of neuronal architecture, therefore, represents a transition from cellular dysfunction to large-scale structural degeneration within the brain. As dendrites retract, synapses disappear, and axons fragment, the physical network that once coordinated neural activity begins collapsing. This structural destruction sets the stage for the next phase of tau-related neurodegeneration, in which the loss of connectivity between brain regions leads to widespread disruption of functional neural networks. Network-level collapse. As structural destruction spreads through neural populations, the consequences begin emerging at the level of large-scale neural networks. The brain operates through coordinated communication between multiple regions that exchange information continuously through synaptic and axonal connections. These connections allow sensory signals, memories, emotional responses, and motor commands to integrate into coherent patterns of activity. When tau pathology dismantles the structural architecture supporting these connections, the synchronization required for network communication begins deteriorating. One of the first networks affected by this disruption is the memory circuit centered around the hippocampus. The hippocampus receives processed sensory information from multiple cortical regions and organizes that information into stable memory traces. This function requires rapid and precisely timed communication between hippocampal neurons and widespread cortical areas. When tau pathology damages axons and synapses within this network, the timing of signal transmission becomes irregular. Signals arriving from cortical association areas may reach the hippocampus with reduced strength or delayed timing due to disrupted axonal pathways. Even when neurons remain capable of generating electrical impulses, the physical connections required to transmit those impulses efficiently have begun deteriorating. The hippocampus can no longer integrate incoming signals with the coordination required for reliable memory encoding. Communication between the hippocampus and the prefrontal cortex also becomes impaired. The prefrontal cortex relies on hippocampal input to support reasoning, planning, and decision-making. These cognitive processes require synchronized activity between distant neural populations connected through complex white matter pathways. As tau pathology disrupts these pathways, the coordination between hippocampal and prefrontal regions weakens. This weakening produces noticeable cognitive effects. Tasks that require integrating memory with current information become slower and less accurate. Decision-making processes may require additional time because signals must travel through fewer or less efficient pathways. The brain attempts to compensate for this disruption by increasing the activity of remaining circuits. But these compensatory mechanisms cannot fully restore the original speed and precision of network communication. Networks responsible for emotional regulation also experience disruption. The amygdala processes emotional stimuli and communicates extensively with the prefrontal cortex, which helps regulate emotional responses and behavioral reactions. Under normal conditions, signals traveling between these structures allow emotional responses to be modulated according to context and experience. When tau pathology damages the connections between the amygdala and prefrontal cortex, the regulatory influence of the prefrontal cortex becomes weaker. Emotional responses generated within limbic structures may become less controlled, producing behavioral changes such as impulsivity, irritability, or reduced emotional flexibility. These symptoms often appear during early stages of certain tau-related neurodegenerative disorders. Attention networks distributed across the parietal and frontal cortices also become affected. These networks depend on synchronized electrical oscillations between distant brain regions. Neurons within these circuits must fire in coordinated patterns to allow efficient transfer of information across the network. The timing of these oscillations ensures that signals arriving from different regions are integrated properly. Tau-induced structural damage interferes with the ability of neurons to maintain synchronized firing patterns. The loss of synaptic connections alters the balance between excitatory and inhibitory signaling within neural circuits. Some neuronal populations may become less active due to synaptic loss, while others may fire irregularly because regulatory feedback loops have been disrupted. The resulting desynchronization weakens the oscillatory rhythms that normally coordinate neural communication. Brain regions that once activated simultaneously during cognitive tasks may begin operating independently. This loss of synchronization reduces the efficiency with which information can be shared across the network. White matter degeneration further accelerates the collapse of network communication. Long axonal pathways that once transmitted signals between distant brain regions become fragmented as tau pathology spreads. Signals traveling through these pathways encounter interruptions where axons have degenerated or synaptic connections have disappeared. Even when neurons remain alive in isolated regions, their ability to participate in coordinated network activity becomes limited by the absence of intact communication pathways. Signals generated within one region may fail to influence activity in another because the structural connections required for transmission no longer exist. The brain attempts to adapt by recruiting alternative circuits. Surviving pathways may increase their activity to compensate for the loss of damaged connections. However, these compensatory mechanisms place additional metabolic stress on neurons that are already struggling with cytoskeletal instability and mitochondrial dysfunction. As degeneration continues, the number of available alternative pathways declines. Neural circuits that once functioned as integrated systems begin fragmenting into smaller groups of neurons with limited connectivity. Information processed in one region may remain confined within that region instead of influencing activity across the broader network. The collapse of network-level coordination marks a critical transition in tau-related neurodegeneration. The pathology has moved beyond molecular disturbances within individual neurons and now affects the global communication architecture of the brain. Although some neurons remain structurally intact during this stage, the networks that once allowed them to function collectively have become disrupted. The loss of synchronized communication prevents the brain from maintaining the complex patterns of activity required for higher cognitive functions. As tau pathology continues spreading and structural damage accumulates, the brain approaches a biological threshold beyond which recovery becomes extremely unlikely. The next phase of degeneration involves the point at which the loss of neuronal connectivity and the propagation of tau aggregates render the neural network permanently altered. Irreversibility boundary. As tau pathology spreads through interconnected neural circuits, the brain gradually approaches a biological threshold beyond which restoration of normal structure and function becomes extremely unlikely. Early in the degenerative process, neurons possess limited capacity to compensate for damage through mechanisms such as synaptic strengthening, network reorganization, and structural remodeling. These adaptive responses rely on the presence of intact neurons, functional cytoskeletal systems, and viable communication pathways between brain regions. The irreversibility boundary is reached when the structural and molecular damage produced by tau pathology exceeds the ability of these compensatory systems to maintain network stability. At this stage, several biological processes converge to prevent meaningful recovery of neural architecture. One of the most important of these processes is the persistence of neurofibrillary tangles within neurons. These aggregates consist of densely packed bundles of paired helical filaments composed of hyperphosphorylated tau. Because of their highly stable filamentous structure, these tangles resist degradation by the cellular systems responsible for removing damaged proteins. Proteasomal degradation and autophagic clearance mechanisms become overwhelmed by the size and density of the aggregates. As a result, tau tangles remain within the neuronal cytoplasm for prolonged periods. Their continued presence interferes with intracellular transport, mitochondrial distribution, and protein synthesis. The microtubule network that once supported axonal transport cannot be reconstructed while large aggregates occupy the cytoplasmic space required for normal cytoskeletal organization. At the same time, tau pathology continues propagating between neurons through mechanisms involving the transfer of tau aggregates across synaptic connections. Small aggregates released from affected neurons enter the extracellular environment and may be internalized by neighboring cells through endocytic pathways. Once inside a new neuron, these aggregates act as templates that promote the misfolding of soluble tau molecules. This templated interaction allows abnormal tau structures to promote misfolding of soluble tau within neighboring neurons. The process resembles the propagation mechanisms observed in certain protein misfolding disorders. As newly misfolded tau molecules join existing aggregates, the filament network within the neuron expands. The spread of tau pathology follows the anatomical organization of neural networks. Neurons connected through synaptic pathways become sequentially involved as aggregates move from one cell to another. Brain regions that share strong connectivity often develop tau pathology in predictable patterns, reflecting the transmission of abnormal tau species along established communication routes. As increasing numbers of neurons become affected, structural damage accumulates across large portions of the brain. Synaptic loss, dendritic retraction, and axonal fragmentation occur in multiple interconnected regions. The network-level compensation that initially allowed surviving circuits to maintain function becomes increasingly difficult to sustain. Another major factor contributing to irreversibility involves neuronal cell death. Neurons subjected to prolonged metabolic stress and cytoskeletal disruption may activate internal signaling pathways that initiate apoptosis. During this process, the neuron undergoes controlled dismantling of its internal components. The cell body shrinks, nuclear DNA fragments, and the remaining cellular structures are removed by microglial phagocytosis. Unlike many tissues in the body, the adult human brain possesses extremely limited capacity to replace lost neurons. Neural stem cells exist within restricted regions, such as the hippocampal dentate gyrus, but their ability to generate fully functional replacement neurons, capable of integrating into complex cortical networks, remains minimal. When large populations of neurons die, the connections they once formed disappear permanently. The loss of neurons also alters the surrounding immune environment. Healthy neurons release molecules that suppress excessive activation of microglial cells. When neurons disappear, these inhibitory signals decline. Microglia remain in a chronically activated state, producing inflammatory mediators that further destabilize the surrounding neural tissue. White matter pathways connecting distant brain regions also undergo permanent disruption during this stage. Axonal degeneration eliminates the physical conduits required for long-distance communication between cortical and subcortical structures. Even if some neurons remain viable within isolated regions, the pathways that once allowed them to coordinate activity across the brain have been destroyed. Reactive astrocytes accumulate in regions where extensive neuronal loss has occurred. These cells produce extracellular matrix proteins that stabilize the damaged tissue and isolate areas of degeneration. However, this glial scar formation creates a dense structural barrier that prevents axonal regrowth across the affected region. New axonal projections cannot easily extend through this environment to reconnect separated neural circuits. As these processes converge, the brain transitions from a state of progressive degeneration to one defined by permanent structural deficits. The circuits that once supported complex cognition and behavior cannot be reconstructed because the neurons and pathways required for their operation no longer exist. This transition marks the biological point at which tau-related neurodegeneration becomes irreversible. Before this threshold is reached, interventions aimed at restoring tau balance or stabilizing microtubules may still preserve neuronal function. Beyond this point, however, the structural foundation necessary for normal network activity has been permanently altered. The final stage of the disease reflects the long-term consequences of this irreversible loss of neuronal connectivity and structural stability throughout the brain. Stable aftermath. After the irreversibility boundary has been crossed, the brain enters a long-term state defined by permanent structural and functional alterations within its neural networks. The molecular events that initiated tau pathology may slow in intensity, but the architecture of the brain has already been fundamentally reshaped by the loss of neuronal connections and the accumulation of persistent protein aggregates. Neurons that survive this process continue functioning within an environment that no longer resembles the original network architecture. Many of these neurons contain intracellular neurofibrillary tangles composed of aggregated tau filaments. Although the presence of these tangles does not immediately destroy every neuron that contains them, they continue interfering with intracellular organization and metabolic efficiency. Axonal transport remains impaired within these surviving neurons. The microtubule system that once provided stable pathways for transporting vesicles and organelles cannot fully recover while tau aggregates occupy large portions of the cytoplasm. Motor proteins responsible for moving cargo through the neuron must navigate an intracellular environment crowded with filamentous protein structures. This disruption affects the distribution of mitochondria throughout the neuron. Mitochondria are responsible for generating ATP required to maintain membrane potential, synaptic signaling, and protein synthesis. When mitochondrial transport becomes inefficient, regions of the neuron with high metabolic demand may receive insufficient energy supply. The surviving neural circuits therefore operate under conditions of reduced metabolic resilience. Synaptic density across affected brain regions remains significantly reduced. The dendritic spines that once supported thousands of synaptic connections have disappeared during earlier stages of degeneration. Each lost spine represents the removal of a communication channel between neurons. Even when neurons remain alive, their ability to participate in complex network activity becomes limited by the reduced number of synaptic contacts. The hippocampus often exhibits some of the most pronounced long-term consequences of tau-related degeneration. This structure normally coordinates the formation and retrieval of memories by integrating signals from widespread cortical regions. When tau pathology has damaged hippocampal neurons and their connecting pathways, the efficiency of memory encoding declines. New information may not be integrated into long-term memory with the same reliability as before. Cortical regions responsible for executive function also remain affected. The prefrontal cortex relies on extensive communication with other cortical and subcortical structures to support planning, decision-making, and behavioral regulation. When tau pathology has eliminated many of the axonal pathways connecting these regions, the remaining circuits must operate with reduced connectivity. Signals that once traveled through multiple parallel routes must now pass through a smaller number of surviving pathways. White matter pathways connecting distant brain regions frequently remain partially degraded during the stable aftermath. Axons that fragmented during earlier stages of degeneration cannot regenerate across long distances within the adult central nervous system. The absence of these pathways limits the ability of surviving neurons to coordinate activity across large neural networks. Although the rapid progression of degeneration may slow during this stage, the biochemical environment within affected brain regions remains altered. Microglial cells often remain partially activated as they continue monitoring and clearing remnants of damaged neurons. Low levels of inflammatory signaling may persist within these regions, maintaining conditions that differ from those present in healthy neural tissue. Neural plasticity allows surviving circuits to adapt to some extent. Remaining synaptic connections may strengthen as neurons attempt to compensate for those that have been lost. Alternative pathways may assume partial responsibility for functions once carried by damaged networks. However, this compensatory capacity remains limited by the number of intact neurons and the availability of structural pathways connecting them. The long-term consequences of Tau Isoform Imbalance, therefore, extend far beyond the initial molecular disruption that triggered the disease process. What began as a subtle alteration in the balance of tau isoforms gradually destabilized the microtubule network that maintains neuronal structure. This destabilization allowed tau molecules to detach from microtubules, become hyperphosphorylated, and assemble into aggregates that disrupted cellular organization. As these aggregates accumulated, they interfered with axonal transport, mitochondrial distribution, and synaptic stability. Neuroinflammation and oxidative stress amplified the damage, while tau aggregates propagated through neural networks along synaptic pathways. Over time, dendrites retracted, synapses disappeared, axons fragmented, and neurons died. The brain that remains after this process is not entirely inactive. Neurons continue firing, circuits continue transmitting signals, and some cognitive functions remain preserved through adaptive reorganization. However, the original structural framework that once supported rapid and coordinated communication across the young brain has been permanently altered. Tau proteins that once maintained the stability of microtubules have become agents of cytoskeletal collapse when their regulation was disrupted. The imbalance of tau isoforms initiated a cascade of molecular and structural changes that gradually undermined the integrity of neural networks. The stable aftermath represents the long-term biological state that emerges once these processes have completed their destructive course. The surviving brain operates with reduced connectivity, diminished structural resilience, and persistent protein aggregates embedded within neuronal cytoplasm. This is Pathology of Fear. And this fear is real.
Pathology of Fear is a medical documentary series presenting clinically documented neurologic and pathological mechanisms for educational analysis.